Hello, and thanks for joining us today for the latest Technology Networks webinar, Maximize Your Sample's Full Potential for Genomic Analysis. I'm Dr. Kate Harrison, Senior Science Writer for Technology Networks, and I am here to moderate today's event. I am pleased to have Dr. Benjamin Thieck and Dr. Heinz Opitz joining us today as your presenters. A warm welcome to both of you. Benjamin joined Miltenyi Biotec's R&D department in 2020. As a team coordinator, he is responsible for various product developments related to Miltenyi Biotec's cancer portfolio. Before joining Miltenyi Biotech, Benjamin was working as a research assistant at the Fraunhofer Mewis and RWTH Aachen University, Germany, where he obtained his PhD. He was working on nanomedicine-based drug delivery to tumors, 3D printed tumor models, and developed image analysis algorithms. Heinz is product manager for sample preparation at Miltenyi Biotec. He received his PhD in biology from the Heinrich Heine University in Düsseldorf, Germany. He conducted his PhD research at the Children's Hospital in Düsseldorf, where he established in vivo studies driving advances in the treatment of high-risk childhood acute leukemia. Before joining Miltenyi Biotec as a product manager, Heinz was a scientific manager in the field of tumor genetics, establishing advanced molecular diagnostics for targeted therapies. So, without further ado, I will now hand over to Benjamin and Heinz.
Yeah, thank you very much for the kind introduction, and welcome everyone to our today's webinar. It's going to be very interesting if you are doing genomic analysis. And when we are talking about genomic analysis, we particularly have also the single cell and single nuclei RNA sequencing in our minds, which offer incredible insights into cellular heterogeneity, but it also represents a lot of significant challenges. Challenges that are listed here, and I'm sure you are familiar with a couple of those challenges. And also those challenges are somehow interconnected to each other. That means if you have a sample that has a low viability, it can have a negative effect on the RNA quality, which will create technical noise, for example, making cell annotation complicated and, in the worst case, wrong interpretations. So those factors can have a very negative effect on your whole results, which could ultimately lead to a repetition of the whole experiment, which will then have a negative effect on the cost of your experiment. And all those points are also interconnected to a very important topic, which is the sample preparation. If the sample preparation is done right, you can avoid all those key challenges. And we at Miltenyi Biotech can offer a lot of products and tools to make the sample preparation for your RNA profiling as best as possible. All right, and what you see here is our agenda for today. I am going to talk about the storage solutions first, and then I will hand it over to Benjamin, who is going to talk about the different tissue types and show you all the tricks. So let's talk about storage solution and why is storage important to mention? Well, the storage could also describe the beginning of the journey of your sample. When we think about a human tumor sample, the tumor sample gets cut out in the clinic. And from there on, it needs to be transferred to the laboratory. So you can already here think about the right solution to do the shipment of your sample. And there are storage solutions that help to prevent negative impact on the viability or genomic landscape. And the other positive effect of storage solutions is you can store your sample overnight or even over weekend. It also depends on when you expect the sample. For example, in some cases, the sample can be shipped to the laboratory at a very late stage, where you don't start with a dissociation. But that's not a problem if you have the right storage solution to store it overnight and then start straight away on the next day. Also, with regards to storage, you might think about long-term storage. And this is maybe because you want to collect a certain amount of samples before you want to start with the whole NGS experiment. And the long-term storage can be done before dissociation or after dissociation of your cells. So, but here I have a couple of informations later on. What can Miltenyi Biotech offer for you in this regard? So when we talk about tissue samples or even mouse organs, we have the max tissue storage solution. And I'm going to show you now some internal data that we got with tissue storage for up to 48 hours at 4 degrees. So we have used as a sample CT26 tumors. We checked the viability after 24 and 48 hours, and we see that even after 48 hours, the viability is still in a very great range. We looked at cellular composition after dissociation of those tissue types, of those tissue samples, and see there's actually barely effects visible on those cellular compositions compared to the fresh sample. And very important, when we look at stress-related genes, after 24 and 48 hours, in most of the cases, we see no or very low effects on the expression of those genes. So this is the solution if you consider storage or shipment of your tissue or organs. But what about if you have already cells in suspension? For this, we have developed the max cell storage solution. And following, I will show you some internal results from a sample for up to 72 hours at 4 degrees. So here as well, we checked for viability, and we see that even after 72 hours, our viability is still in a very good range. Cellular composition looks also great in this regard, even after 72 hours. And here as well, we had a look at the transcriptome of the cells. Especially, we have done here a whole exome transcriptation analysis. And we have even compared our cell storage solution with the competition that we call here medium HT. And we see that the transcriptome is less affected throughout the storage time points in comparison to the medium HT. And we even had a look at the cellular stress response genes. And we see as well here the same pattern with the max cell storage solution. We see a more stable transcriptome in comparison to the medium HT. We also had a look at the functionality and especially to specific cell types. So we have isolated monocytes and T cells and checked the viability after 72 hours. Both look very great. And we did functionality assays where we have used the monocytes and generated monocyte-derived dendritic cells. We saw no differences in comparison to fresh cells. And also with the T cells, we have done proliferation assays. After 3 days, the results look quite great. And we even have done analysis after 6 days, but this is what we don't recommend. So the cell storage solution for 3 days is okay. All right, so now we have talked about the short-term storage. What about the long-term storage? For this, we have a solution as well. It is called the max freezing solution. And with this internal results, we have done some experiments on PBMCs dissociated CD26 tumors and CD26 tumor pieces. And here we see as well great viability with PBMCs, tumor cell suspensions after thawing. What we see with tumor pieces is that the viability is quite reduced when we compare it to tumor cells in suspension. This is just simply because the freezing solution can cover the whole cell, and which is not true if you want to cryopreserve tumor pieces. That means not every cell is covered with the freezing solution that protects during thawing and freezing. But nevertheless, storage of tumor pieces is also possible. You then need to further remove that cell and debris, and Benjamin will uh, tell a little bit more about this tools. Nevertheless, we have also looked at cellular composition, and we see here as well no significant effects. I also need to say here we have compared our freezing solution with the homebrew method, and what makes our freezing solution particularly interesting is we don't have any human serum or animal-derived compounds in our solution. It is a ready-to-go solution. So whenever you would expect some negative effects that are due to the serum in your freezing solutions, this is not true for our freezing solution because it's completely animal component free. All right. And with this, I would like to hand over to Benjamin, who is going to tell you a bit more about the different tissue types.
All right. So also warm welcome from my side. So now after having Heinz presented you our solutions for short or long-term storage, I will pick up the line of thoughts and talk you through some of the sample preparation workflows with different tissue input materials. So these workflows are, in our opinion, the best practices for fresh, frozen, and FFPE tissues if you want to perform NGS applications. Starting off with fresh tissue. So once you have your fresh tissue in the lab, you need to dissociate it if you want to perform single cell sequencing. And we have the GentleMax technology and associated tissue dissociation kits, which you can use to dissociate the tissues. Afterwards, you should clean up the sample in case you are interested. You can isolate target cells of interest. You should, of course, QC your cell suspension so that you know and are sure that you are passing some quality criteria for the downstream sequencing application. And I want to focus a bit more on this cleanup step because here we have a new solution, which is actually a simple one-step cleanup protocol, our cell suspension cleanup kit. So on the left, you actually can see images of the sorry. So now, here on the left, you can see an image of a sample which was dissociated but didn't receive any cleanup. And on the right, you have the image after using our cell suspension cleanup kit. So if you have a closer look at a part of the image, you can see that on the left, you actually have green, red, and grayish circles. So this is actually an indication for live, dead, or erythrocytes. And on the very right, you don't see that mixture anymore. So the dead cells are removed and also most of the erythrocytes are gone. And this is actually also supported by the flow panels you see beneath. So where you have PI as well as glucophorin A expressed events here on top on the pre-cleanup sample, which are almost gone in your sample after you have performed the cleanup. And how does it look in numbers? So with respect to viability, when we have dissociated these fresh human ovarian cancer samples, we could increase the viability from, let's say, a bit more than 50% to almost 80% on average, while reducing the RBC content to a level of less than 10% on average. At the same time, which goes, let's say, hand in hand with the RBC content, we increase the staining efficiency, which is just the number of nucleated cells you are measuring. And also nicely, while performing the cleanup, you are also removing free mitochondria, which are in your solution after you have dissociated the tissue. So what impact actually does it have on your sequencing if you have a clean sample or a sample which wasn't cleaned before sequencing? So there are some general QC metrics, for example, valid barcodes, where you can see that we have an increase of approximately 1.5% of valid barcodes in the sample which was cleaned up using our kit. And that actually means that there were more cells captured, and having more cells captured, there you have also the chance of getting a greater diversity of cell types in the sample, and you can have a closer look and eventually also have a more detailed biological insight into your sample. So in the end, it is actually adding more data points for analysis. So the next metric are reads mapped confidentially to the intergenic region, which is more or less, let's say, a measure of how much noise you have. You want to have your reads mapped to the transcriptome because you're interested in which genes are actually expressed. So this kind of gives you information of how much, as mentioned, of how much noise you have and how much, let's say, also reads are lost into regions which you are not interested in. And furthermore, we have here reads mapped confidentially to the transcriptome. So this is really what matters in the end, right? So this is what it's all about in single cell RNA sequencing. Reads mapped to the transcriptome. And here you can see that we have a quite clear increase on average of approximately 8%, which you can then interpret as you have higher data quality, you're getting more meaningful data, or the transcriptome is covered better, and also just overall you had a more efficient sample preparation. So you are increasing the experimental efficacy by higher data quality with minimal noise. And these are general QC parameters, which you are getting after your sequencing run using the 10x platform from Cell Ranger. So I also want to show you the impact on one level beneath if you're performing your analysis in, for example, the Loop browser. So we did actually a cell calling, not the default cell calling, but a forced cell calling to a theoretical cell count, which was in our one sample, which wasn't cleaned up, which resulted in 16,470 barcodes, whereas we have in our cleaned sample 14,987 barcodes. However, what is interesting is, so you can select a threshold of mitochondrial UMIs per barcode. So mitochondrial UMIs are an indicator, for example, also for apoptotic cells. So it's often done that one puts a threshold around 15% to remove barcodes with such a high mitochondrial UMI content. And as you can see on the left, there's actually 25 or more than 25% of barcodes which you are removing, whereas there are only 5% of the barcodes removed in the clean sample. And this again is is also reflected kind of then in your reads, right? So there are a lot of reads which you are filtering out here at that step, which are not useful data anymore. So you're not using them on some regions which are of interest and relevance, but they are kind of trapped in some, let's say, data which you are filtering out later anyhow. And this is not only true for mitochondrial reads, so you have some UMAP plots here where I have indicated or highlighted the barcodes or cells where we still detect hemoglobin gene expression. And on the left, you do see that there are around 600 barcodes where we still detect an HBV gene expression, whereas on the right in the clean sample, you just have 4 left. And again, these are reads which get trapped in events you are not interested in. And of course, that reduces the efficacy, also monetary efficacy of your experiments if you do not perform this cleanup. So certainly there are other ways to perform cleanup. So mostly, I think most well known are those 2-step cleanup workflows where you do an RBC lysis and a dead cell removal. And of course, we also wanted to benchmark our cell suspension cleanup kit against this 2-step procedure. And as you can see, we have again an increase in viability, a quite high increase in viability and reproducible increase in viability when we were using the cell suspension cleanup kit, whereas when we were performing this 2-step procedure, we were only able in 2 out of 3 cases to just just surpassing the 70% viability threshold, which is given by 10x. And the same is then also true for the RBC content. When we were using the cell suspension cleanup kit, we could quite nicely reduce the RBC content below 30%, which is also a threshold given by 10x. However, using the hypotonic lysis, we were only able to get below 30% in 1 out of 3 cases. And of course, the hypotonic lysis and also the increase fraction of dead cells in the end is most likely resembling this lower RIN we have detected, so the RNA integrity number. Unfortunately, we don't have sequencing results ready yet, but they will be presented in an upcoming app note. So increasing reproducibility and sample quality while saving 30% hands-on time is possible when you are using this 1-step cleanup protocol, which we have released with our cell suspension cleanup kit in comparison to the conventional 2-step procedure, which is often promoted by competitors. All right, so coming to the next tissue type, so frozen tissue, extraction of nuclei and their purification. So we start off, when we're looking at the workflow, we are starting off with the tissue. It can be fresh, snap frozen, or OCT embedded tissue. Put it into the C-tube, add the nuclei extraction buffer and inhibitors, put it onto the GentleMax and the GentleMax octocooler, run a really short nuclei program. You can filter the suspension. You need 1 centrifugation step, another filtration, and you're good to go for sequencing. So as already mentioned, just as an example, you can use fresh or frozen tissues or other, as mentioned, OCT embedded tissues for this workflow. And the input material can range from 250 milligrams, but can go down to 25 milligrams or even less. And you are still able to isolate quite a high number of nuclei. And it's not only that you can isolate nuclei from diseased tissue, but also from healthy tissues like brain, liver, heart, or kidney, and then diseased ones, melanoma, breast cancer, colorectal, prostatic cancer, and again, also with a wide variety of input amounts. You are able to extract a lot of nuclei for your sequencing experiments. So it's one thing to extract the nuclei, but you can imagine that there are still a lot of debris produced which is surrounding the nuclei, which might also be still in your cell suspension or nuclei suspension. Therefore, after extraction, we also offer the solution to actually enrich the nuclei. So once you have extracted them, you spin them down, resuspend them in our antinucleus microbeads, and enrich them via a magnetic field and a LS column. The other option would be to do cell sorting, for example, using the Max Quantito. Why to do so? Why should I do that? And here you can see the why. Because depending on the sample, the input sample, there is quite a lot of debris. Maybe not so much for human ovarian cancer samples as you have them here on the right, but if you are looking to the other samples on the right, you can see that especially for brain samples, there is a very low purity of the nuclei after the dissociation, which requires an enrichment to purify the nuclei for your to also make the sequencing afterwards more efficient. And more details on that, how does it look? So the brain samples here on the very right after dissociation are looking quite crude. So you see a lot of debris around the nuclei here on those two images. You have a purity here in this flow plot of 5% before enrichment, but after enrichment, you can increase the purity really to 87% in that example. And you can appreciate here on these images that they are looking quite clean. Having a closer look now at the nuclei itself and also comparing the shape of the nuclei with some example image from the 10x Genomics website for quality assessment of nuclei, you can really see that our nuclei, which we have isolated here from this frozen mouse brain, is really perfect in shape and meets the highest quality standards, as you can see here on the very left side of this image from 10x. How does it impact your sequencing? Having a really clean sample which you fed into the NGS workflow. So if you're looking at these barcode rank plots, first of all, the UMI counts, it's not very obvious, but it's a one magnitude higher in comparison to the non-enriched mouse brain sample. And also if you're looking at the cliff or the knee true features of this barcode rank plot, there's quite steep cliff and a pronounced knee in the enriched samples, whereas the cliff and the knee are not that steep and not as pronounced in the non-enriched brain sample. And having it pronounced is also, again, a measure of a good sample quality. This you can see here on the next slide. So you have an increase in the fraction of reads in the enriched sample. You have clearly a decrease of mitochondrial reads in your sample. So you have a clean nuclei fraction, which you are sequencing in comparison to the non-enriched one. Then on top, you have a significant increase in the median genes and UMI counts in the enriched samples in comparison to the non-enriched ones. And you do also see a good separation of the individual clusters here in the UMAP plot. So overall, it's really increasing the efficacy of the experiments you are performing when you are using really a nice and clean input material. So coming to the last topic for today, FFPE tissues. So also here, I want to first of all give you a brief overview about the whole workflow. So it's actually starting off with the FFPE tissue, which you can then cut off some curls, 25 to 50 microns, put them into the seed tube, perform deparaffination and rehydration in the seed tube using the very same seed tube, adding enzymes, dissociation enzymes, which are actually now part of our FFPE tissue dissociation kit for RNA profiling, which we have released this month, and dissociate those samples on our GentleMax. Afterwards, you perform a filtration, you count and QC your sample before then going for this NGS workflow. So having a look at the demonstrated protocol revision D by 10x, as I've mentioned, we have this deparaffination and rehydration process in the C tube, and then you're adding the dissociation mix and perform the dissociation on the GentleMax octal. And this dissociation mix here in the demonstrated protocol is Liberase TH. And so we have set up or developed this new FFPE tissue dissociation kit for RNA profiling, where we have really selected some enzymes which are perfectly suited for sequencing applications, for downstream sequencing applications, which are superior to this Liberase TH based dissociation. And I want to show you some of the results which we have obtained during our developments. So we took actually here FFPE breast tumor blocks. We cut 2 × 25 micrometer scrolls, and now you can see the barcode rank plot. You have the barcode rank plot in which is slightly lighter, which is actually coming from the Liberase TH-based dissociation, and then the bold line, which is coming from the dissociation which was performed with our new FFPE tissue dissociation kit for RNA profiling. First of all, you see also here a high UMI count, a steeper cliff, and also a more pronounced knee. In these FFPE samples, these are not as pronounced as in fresh samples, but this is known and this is normal. Looking at the QC parameters, I've just listed all of them, but want to highlight 3. So median UMI counts per cell, median genes per cell, and median reads per cell. And in all cases, you actually see that we are obtaining twice the amount you would obtain using the Liberase TH-based dissociation. And we haven't tested it only on FFPE tissue like breast tumors, but also on several other tissues. And in all cases, we obtain superior results in comparison to the Liberase TH based dissociation. So we have an improved single nuclei RNA sequencing performance, indicating enhanced data quality and sequencing efficiency. So single nuclei RNA sequencing, why do we think that we are performing that and not single cell? It is also what 10x thinks. So they think also that the majority of reads are actually coming from nuclei and not from whole cells, which were extracted from the FFPE tissues. And also now looking at the mitochondrial gene expression of samples which were either dissociated using the Liberase TH or the FFPE TDK for RNA profiling, we can see a clear reduction in mitochondrial gene expression. So the reduced detection of mitochondrial genes suggests that we are actually having a purer nuclei fraction with minimal contamination from cytoplasmic remnants, which in the end also allows you to have a better interpretation of your data, because you will have less background information coming from eventually something which you are not interested in. Coming back to our workflow, you might ask yourself, is that actually also feasible for sections which I have already mounted on glass slides? And what are their tissue size limitations? And yes, you can actually do so. So we have performed the head-to-head comparison using our new dissociation kit, where we have performed a sequencing experiment where we used 5-micron thick slices as input material. So the deparaffination and rehydration step was done on the slide. Then we removed the sections from the slide and put them into the C tube and made the dissociation with our dissociation kit. Of course, one needs to test that because the efficacy of cells which are recovered is lower in comparison to those thick scrolls. But if you are performing such an initial experiment, you can easily adapt also this protocol to this slide method, and we are really happy to support you to set up these protocol in your labs as well. With that, we are actually coming already to the final slide of today's webinar, and I hope we were able to show you that sample preparation is really able to address all of the hurdles or challenges, or many at least of these challenges you are faced when you are performing NGS applications, and that we here at Miltenyi Biotech offer a lot of solutions to get most of your data so that you can really run your sequencing experiment in a highly efficient manner. And with that, we are done, and thank you for your attention and are happy to take any question.
Thank you very much, Benjamin and Heinz. I'm sure everyone will agree that there were some really excellent points raised during that presentation. We will now begin the Q&A session. And since we have two different speakers, I'm just going to group them by topic. So if we address the FFPE questions first. So the first question we've got is, do you dissociate the FFPE tissue to obtain cells or nuclei?
I mean, I elucidated already partially that within the talk. So also in our discussions with 10x, they were rather saying, okay, there's the larger proportion of the results we are obtaining are coming from nuclei. And we see that as well in our experiments. And for us, it might be even stronger. So if you are dissociating your samples using our kit, most of the reads will be coming from nuclei and not from single cells.
Brilliant, thank you. So another FFPE question we've got is, can you purify FFPE nuclei with the anti-nucleus microbeads?
Would be great, but unfortunately it's not possible. So the anti-nucleus microbeads, they require an antigen retrieval step, which is not part of the FFPE dissociation kit for RNA profiling.
Okay, that makes sense. How does deparaffinization work with your kit?
Okay, so that's actually as it was shown on the demonstrated protocol. So the deparaffination is done in the C tube. So that's actually quite handy that you can directly move your tissue sections or scrolls into the C tube, perform the deparaffination rehydration within the C tube, perform then or add the dissociation enzymes into the C tube at the very end of the process, close it, go to the GentleMax and start your dissociation.
Okay, thank you.
Benjamin,
Oh,
sorry, if I might add something to this,
yeah, go for it.
which could be also very interesting. The deparaffinization is actually as usually, right? So it's done as usual, and the most common method is also to use xylene. But I've heard already from a couple of customers, xylene is pretty difficult to use in a couple of labs. So they asked us already if it's possible with the xylene alternative. And Benjamin, if you maybe have something to this with regards to this.
I mean, it would be great, and we will probably also come up with a solution so that you can use xylene alternatives. I don't have any names now in my head, which you could use as an alternative, but there are already some beta testers or beta tests running where we are testing xylene alternatives, and maybe that will be then part of an upcoming protocol revision in the future.
Okay. And I think the last FFPE question we have at the moment, although if you still have an FFPE question, please go ahead and ask it. You still have time. What is the difference compared to the old FFPE TDK?
Okay, yeah, there's another FFPE tissue dissociation kit, so don't confuse them, because the one which is already at the market, there we are aiming to isolate cells, and there is also the possibility, this is also including an antigen retrieval step. So this is not optimized for downstream NGS applications, so there really pay attention now if you're looking on our website that you get the right kit for the right application.
Okay, thank you very much. Let's move on then. Is nuclei hashing after nuclei purification with microbeads possible?
I think I will take over this question if it's okay for you, Benjamin.
Sure.
So we had this question already a couple of times, and we also had a look at a prominent one. So I hope I remember it correctly. It's a MAB 414, something like that. It could be possible. And we looked at it and we saw that actually hashing is possible. So we have isolated nuclei and then used this particular antibody, and we saw that let's say all nuclei could be stained with this one. I'm sure I've heard about two different antibodies, and yeah, I can look it up and send the results from our hashing experiment if I can get the email address. Yeah.
Cool. Thank you very much. Um, okay, this is interesting. If I am working with zebrafish, does the anti-nucleus microbead do the anti-nucleus microbeads also work for this species?
Yeah, that's a very interesting question. Since we don't have these types of tissue available at our facility, we cannot say that we have internally validated it, but I'm sure we have a lot of customers, and this is really great if we get the feedback, hey, I've used species XYZ, used your stuff and it worked. So that's awesome because then we can answer this to all the other customers who are asking that the same question. What I can do is I can ask our tech support because they have the library of all the cases that reached us before. And there I can ask if zebrafish was already mentioned. What I can say is that we have already a couple of customers tested it on several species like pig or even C. elegans, and they were able to isolate the nuclei and clean it up with our antinucleus microbeads. It would be interesting if it would not work with zebrafish. Yeah.
Okay, great. I'm sure that would be really useful for our question asker. Um, can I also isolate nuclei from plants?
Oh yeah, that's basically the same kind of question.
Yeah.
Um, yeah, so we have already heard from other customers who are working with nuclei from plants, and actually the antinucleus microbeads seem to work. They have also shared some documents with us, some data that I can also share if I have the email address. That shows that antinucleus microbeads work per se with the plant nuclei. However, nuclear extraction is a different topic. I think plant material needs different type of nuclear extraction buffer. I've heard about the buffer. It's called Honda buffer. So maybe this thing needs to be done with a different buffer, but then afterwards, isolating nuclei should work with the antinucleus microbeads.
Okay, fantastic. And we're just going to have one more question now because we are starting to run out of time. So, don't worry if you haven't asked your question yet or you hadn't had your question answered, we will get you a response offline. So the final question is, how does cryopreservation affect the outcome of single-cell sequencing?
Yeah, that's also a good question. And Benjamin, correct me if I'm wrong, but what we have done is we tested our freezing solution on tissue chunks and also already dissociated cells and then cryopreserved the cells and have done then sequencing. I think it was also the single cell sequencing. What we could see is that the quality of the outcome is a little bit better when you dissociate your cells prior to cryopreservation. So this is what we have seen first. And the other thing is it is per se possible, but I would not compare it to fresh material because fresh is a different level of quality. So whenever fresh is possible, go for fresh, but cryopreservation is can be, of course, considered as well. So we are actually working on a data set. So as soon as this is available, we could also share this for all the customers who asked for this at our technical support.
Okay, fantastic. So thank you very much to Benjamin and Heinz, but I'm afraid that is all we have time for today. Thank you again for your time, and thank you to Benjamin and Heinz for a fascinating presentation today.